3 segments. And so, hey, I will start. I'm going to be the introducer today, but most of you all know me, but just in case you don't, I'm Melinda Riccitelli. I'm the president of the US CCHS network. And I want to thank you all for attending the 4th virtual CCHS research forum. So this is a forum brought to you by the International CCHS patient community. And we started doing these during COVID as a way of, staying connected when we couldn't meet. And we do them twice a year virtually. And we fill in our virtual meetings with an in-person meeting. That last in-person meeting was in Florida, in Orlando in 2023, right? September of 2023. And so the next one we'll have of this will be sometime in the fall. So I did want to start with just some basic rules. We have 3 speakers today. I'm thrilled to be introducing them. But we are recording this is 3 segments, hopefully. But when the speaker is speaking, if you could just put yourself on mute, that would be appreciated. We will have each talk is about 35-40 minutes, and then followed by a question and answer period. You can also, when we enter the question and answer period, it'd be helpful if you used your react button and raised your hand, or somehow, let us know that you had a question. Try not to talk over each other. And then you can also put your question in the chat if you want. And Avatal is going to be monitoring the chat, so you'll be able to, we can ask the questions that way also. So we do have an impressive lineup of speakers today, and I'm thrilled to be able to announce them. And so we're going to start with our first speakers, Dr. Isabella Ceccherini and Dr. Muriel Thoby Brisson. Isabella's from Italy. As most of you know, I don't really need to introduce any of these people. You probably already all know them. And Muriel is from France. And so Dr. Isabella Ceccherini began her work at the Gaslini Institute's Medical Genetics Unit in 1988. And she's currently the head of the UOSD Laboratory of Genetics and Genomics of Rare Diseases at the Gaslini. She's been studying CCHS since 2002. And she's done a ton of work in CCHS, developing protocols and running genetic tests, identifying somatic mosaicism in asymptomatic parents, and exploring pathogenic mechanisms of FOXP2B mutations, as well as testing potential drug candidates for beneficial effects. And so her work is impressive. I really also want to say thank you, Isabella, because you have been a steadfast and supportive friend and ally within the CCHS community. You've always offered your help and encouragement and expertise to the community and families. And so you've really impacted our space. You've helped us raise awareness and promote CCHS. So I've known Isabella for a while. I have to visit you too, Isabella, in Italy. So we're so happy to have her speaking today. Now, her collaborator is Dr. Muriel Thoby Brisson, and she's a researcher at the University of Bordeaux in France. And although Muriel, there she is up at the top, she's been involved in CCHS research for a long time. I only had the opportunity to really meet her last fall in October at the Breathe conference, and where she did an impressive presentation on regulation of breathing and CCHS. So Muriel did her postdoc with Nino Ramirez in Chicago, and then she took a full-time position, again, I'll probably mess this up, at the Champagne Orte lab in Paris. And where she really started to focus on CCHS, especially after the 2008 Dubray paper that established a mouse model for CCHS. In 2010, she started a lab in Bordeaux where she studies central mechanisms of CCHS-associated respiratory deficits. And she is a familiar player in the breathing community. And so today they are going to do a joint presentation on their latest research, which is a histone deacetylase inhibitor called SAHA. And their work was recently published last December in Molecular Therapy and Nucleic Acids. And so please join me in welcoming Isabella and Muriel to lead off this forum. I think Isabella's going first. So she's going to share her screen.
Okay.
And now I'm going to mute myself.
Okay. Can you see my screen?
Yes.
Okay. So, Melinda, thank you so much for your very nice introduction. I did many things on the CCHS field, as you said, but I should now mention all the people who collaborated with to these achievements. And I cannot list all of them, but just let me mention Tiziana Bacchetti, who shared more than 20 years of CCHS research with me. So and I forgive me for not mentioning all the others, because for the sake of time, of course, because I don't want to take time to Muriel, and I really hope that I will be on time on this first part. But in case, please, in 20 minutes, let me know that my time is over. Hello everyone. First of all, I would like to thank the organizer of this forum, also on behalf of Dr. Muriel Thoby Brisson, who will share this presentation with me. And I want to thank you for your interest in our work and for the invitation. We will present the research approach we have applied to the identification of molecules with potential therapeutic effects on CCHS symptoms. And I will start with an introduction and the in vitro work, while Muriel will conclude with the ex vivo approach. And as you well know, central congenital hypoventilation syndrome is a rare genetic disease caused by defects of the FOXP2B gene. It is characterized by life-threatening breathing deficits, and no cure is available today. And of the nearly 150 FOXP2B variants we have detected in the last 22 years in our institute, nearly almost 80% were polyalanine repeat mutations, and the remaining were non-polyalanine repeat mutations, and with the minority of polyalanine contraction. And these different FOXP2B variants have different frequencies, but have also different impacts on CCHS manifestation and outcome. And here you can see the effect of the transfection of a FOXP2B polyalanine expansion construct in cells. And the construct we have used in this case corresponds to the largest polyalanine expansion. And you can detect the green signals inside the nuclei, in the cytosol, and in both the cellular compartments, both as aggregates and as diffused signals, and this is something we had known for a long time now. If the expression of a large polyalanin expansion inside in vitro in cells induce aggregates formation and mislocalization of FOX2B proteins, and these effects are length dependent, and so they are they worsen as the extension of the alanine increases, and we can say, as you can see in this graph, of course, and so cytoplasmic retention with aggregates can be considered as a reliable marker of the effects of FOX2B polyalanine expansion in vitro. Aggregations occurs when proteins are misfolded, and they are misfolded when they are mutated or overexpressed, and also when the autophagy and the proteasome degradation systems are overloaded, so that they exceed exceeding their ability to eliminate misfolded protein. And in this process, also chaperone-mediated folding has also a crucial role. So protein aggregation and toxicities are increased by autophagy and ubiquitin proteasome system inhibition, and are reduced by HSP70 upregulation and HSP90 regulation. So each shock protein 17 upregulation and each shock protein 19 inhibition. And we have known for a long time geldanamicin, which is a molecule, an antibiotics, which is an inhibitor of HSP90, thus upregulating HSP17. It's the effect of increasing dose of these antibiotics in transfected cells is exactly what expected on the basis of what I've just said. And so GA, or geldanamicin, increases the nuclear localization of FOX2B and decreases the aggregate formation. So it's not only a GA, but it is also a less toxic derivative of GA, which is the 17-AAG, that are both labeled to induce aggregates degradation and restoration of nuclear FOX2B localization. And for these two molecules, this happen by inducing protein quality control and regulation of its shock protein expression. And last but not least, we already knew that the GA and 17-AAG are able to recover some of the transcriptional function of FOX2B, which is lost when FOX2B is mutated, and in particular when FOX2B has a very large polyalanine expansion, like it is shown in this graph. So polyalanine expansion can lead to aggregates formation, cytoplasmic retention, loss of transcriptional function. So, and we wondered whether FOX2B aggregates can be targeted to treat CCHS. And in order to answer this question, we have applied the transcriptomic approach, which is the analysis of all the RNA transcribed in a biological sample at a given time. And these provide insights into levels of gene expression, answering questions regarding a disease. our findings were recently published in a paper that we will present today. And this is the paper. And let me acknowledge all the co-authors of this really gratifying collaboration that we had all together. And we began by transfecting in triplicate a neuroblastoma cell line with wild type and polyalanin expanded FOX2B construct with or without 17-AAG treatment. And all the genes are capable of discriminating the expression profiles induced by treated and untreated polyalanin expanded constructs were identified. And you see these two expression profiles are really different and well discriminated by these genes. Each line is a different gene, and after the identification, they were subjected to gene network reconstruction by using a gene set enrichment analysis and the cytoscape. And in both cases, we found a number of pathways that were already known to contribute to the FOX2B mediated CCHS pathogenesis, like protein aggregation, 70-AAG's response, and so on. But we also found other pathways that are being confirmed, whose involvement is being confirmed in vitro by us. So the treatment with 17-AAG of cell expressing the largest polyalanin expansion is able to rescue a normal gene expression profile. However, geldanamicin and 17-AAG are toxic. So we at this point we wonder whether there are other molecules with similar effects but less toxicity. And to do this, we sought to predict drugs that may have a beneficial effect in CCHS, starting from this, the data I just showed you, the differential gene expression data. And to this end, Nathan Bekush have queried, and his collaborator have queried the connectivity map, which is an online software able to examine the connection among diseases, genes, and drugs through a very large collection of compound-induced reference gene expression profiles. In other words, what we did, or what Nathan did, was the identification of compounds having an RNA sequencing signature similar to 17-AAG. And also to, among all of these compounds, to look at a dose with a clinical relevance. And we then pre-selected the 4 molecules with putative beneficial effects, and these are for drugs. And we also selected, you see, this is the list of the most similar compounds. I mean, the compounds that have a signature, an RNA signature most similar to 17-AG, and these are the compounds that have an RNA signature least similar to a 17-AG. So we also pre-selected 2 molecules expected without an effect, the one with this asterisk. And then we underwent their validation. And Chiara Africano, the first author of the paper I showed you, did all this job. And first of all, we tested the recovery of nuclear localization of mutated FOX2B after the treatment with the 6 molecules pre-selected, finding that the 4 putative beneficial molecules had a FOX2B localization similar to the one shown by the geldanamicin treated cells, while the 2 no effect molecules had behaved like the untreated polyalanin expanded expressing cells. Exactly as we expected, of course. And after that, we tested the effect of selected drugs on the FOX2B mediated transactivation of the DBH promoter, finding that the FOX2B plus 13-ALA has almost 3 times or more than 3 times fold reduced activity compared to the wild type. And while the treatment with GA gave the expected results, rescuing most of the lost activity, and SA 10 millimolar was the molecule and the dose that had the best effect among all the drugs. So SA or vorinostat showed the best efficacy and the less toxicity. And this molecule at this dose was further tested in vitro for its effect on FOX2B aggregates and ex vivo for its effect on breathing parameters. And so the effect of 10 millimolar SA was tested on FOX2B aggregates function, of course, finding first of all what we expected. So a prevalent nuclear cytoplasmic localization for the expanded FOX2B compared to the wild type, and also the induction of a large number of aggregates per cell. But once the cells were treated with SA, the FOX2B was relocalized in the nucleus at a significant extent, and also the aggregates decreased a significant extent. And the SA was also able to recover part of... Pardon? Yes? Okay. And SA was also able to recover part of the luciferase activity. And last but not least, we also tested the effect of the 7-ALA expansion in vitro. 7-ALA is the most frequent PARM in CCHS patients, and is also the mutation that is carried by the animal models that you'll see later. So on cell culture, SA is able to partially eliminate FOX2B aggregates, significantly restore FOX2B nuclear localization, improve the regulation of FOX2B target gene expression, and now we will see which the effect of SA is on breathing activity. So I leave the screen and the podium to Muriel. Thank you very much.
Isabella, can you stop your sharing your screen that I can take over?
Yes, I'm trying. Yes, done. Okay, thank you.
Thank you. Okay, can you see my screen? Yes?
Not yet.
Not yet?
You have
to see that you're trying to share.
But you don't have the image up. You don't have your PowerPoint up yet.
Okay, but I don't see my screen actually.
Maybe you should try to reshare it. Stop and reshare it again.
Okay. So I stop and I reshare. No, not this one. Is it better?
Nope. We know that it worked out. We tested it.
Fine?
Not yet, but it worked for you at the beginning. So it should
Oh.
work.
Sorry for that.
No problem.
So Muriel, you could send it to Avatar or I and we could share. If that's easier. I don't know how big your presentation is.
Now
Okay.
it will
Yeah,
work.
we got it. We got it.
Good.
Yep.
Okay. Okay. Like this. Yes?
Just put on presentation mode. Yes.
Yes, it is. Is it?
Yes.
With a pointer.
Good.
Okay, sorry. So now, actually what I did in this project was to test the different drugs on respiratory functions. To do this, we used a well-established animal model for CCHS that has been produced by Christo Goridis lab, and that bears a +7 alanine onto the FOX2B gene. At birth, the animals are cyanotic and they actually exhibit hypoventilation, a lack of response to hypercapnia, so very typical to CCHS syndrome. And most of them are actually dying very quickly. So these animals is a very good model to test what is happening on the breathing activity. So what do we know about the neural network that are actually constituting the central respiratory command? Actually, there's several groups. The first one is the RTN-PF4G, which is the retrotrapezoid parafacial respiratory group that controls pre-inspiration, expiration, and is a main actor of central chemosensitivity. Sorry, this is in French. I didn't change the name. And most importantly, this group is known to be anatomically and functionally affected in CCHS. The second group is the pre-Bottinger complex that is actually controlling inspiration, and this group is pacing the rhythm. It's doing the rhythm itself, so it's very important. And the third group is the PICO, most recently discovered by Nino Ramirez, that is controlling post-inspiration, but I won't talk about this group today. So we have all the major actors that are involved in creating the breathing rhythm, and all those are interconnected and send their signal to the phrenic nerve that will innervate the most important respiratory muscle, the diaphragms. And actually on those preparations, we can record the electrical activity that is coming out from this nerve as an image of the full respiratory command. So what we are using, we can isolate this part of the nervous system from those animals, put them in a recording chamber and expose them to the different drugs. So that's exactly what we did. We isolate the brainstem with all the components and treated them with the drugs. But because the animals are dying, we harvested each animal and each brainstem just before birth, before it's too late, I would say. Okay? So here you have one example of the type of recording we can do from a wild type animal and also from a mutant animal. And all the peaks you can see here are one signal for one inspiration. So as you can see in the wild type, it's spontaneously active, while in the mutant, there's still activities, but at a much slower frequency that would correspond to the hypoventilation. When we made a challenge by changing the pH to mimic hypercapnia, what you can see on the wild type preparation is an increase in the frequency that was fully expected. And as we know, in the same condition, the mutant animals are completely unable to respond to this acidification. And here you have the quantification of what we found for several preparations with here the mean frequency of the preparations, and in green the wild type preparation, in blue the mutant ones, and you can see a huge difference in the basal frequency, and also that the mutant preparation exhibit almost no response to the pH changes. So this is the same quantification using a different representation. But because we wanted to test a long-term effect of molecules, the first thing we had to do was to verify that this preparation can stay alive for a long time in vitro. That's what we did. And you can see that after almost 8 hours in the recording chamber, the preps are still rhythmically active. They are still responding to acidosis, but the mutant remains unable to respond to pH and are still showing a slow frequency. So the next step was to expose preparations to SAHA, the molecule that Isabella just talked about. And what we found was several things. First of all, the wild-type preparation remained unaffected, which is good. But in the mutant preparation, you can see that the frequency is now much faster, almost similar to the wild-type ones. And still, moreover, we have an increase in the frequency when we change the pH. That would be a kind of recovery of chemoception. So here you have the quantification, and in dark blue you have the wild-type preparation, and in dark green you have the mutant preparation, and you can see that there's now almost no difference between the wild-type and the mutant preparation after more than 7 hours under SAHA. So those results show clearly that, first of all, SAHA is able to induce an increase of the basal frequency. You can see here there's no more difference. And second, there's a restoration, at least partial, of the possibility for the preparation to respond to an hypercapnic environment. So this molecule is very promising, but because we wanted to have, or at least to be able to identify different molecules of interest using different pathways and with different mode of actions, we got interested in a second molecule, which is actually the 17-DMAG. So it's a derivative of 2 antibiotics that Isabella talked about, but that are toxic. So hopefully this one is less toxic. And it is known as being an HSP90 inhibitor. And as Isabella said, all the heat shock protein response pathway is important to get rid of the FOX2B aggregates. So the first thing we did using Western blot analysis, we estimate the quantity of HSP90 and 70 in wild-type preparation and in mutant preparation. And the first thing we found was that it seemed that the HSP 70 protein are less expressed in the mutant. This is quantified here, control and mutant preparation. Then the question is 17-DMAG capable of changing this? Absolutely. After exposure to the drug, you can see that HSP70 is now expressed almost equally compared to the HSP90, and we restore a completely normal quantity of protein. So what we suspect is that this molecule is able, first of all, to upregulate HSP70, but it's also known to be an HSP90 inhibitor, probably not acting on the quantity of the protein, but rather on its activity. Then we decided to check whether this new molecule is also able to improve the respiratory activity. So we used the exact same paradigm than the one I just told you about when we used SAHA. So I will maybe go a little bit faster on this because those are exactly the same recordings. Here you have the wild type preparation, the mutant preparation, and you can see that after 8 hours in the drugs, the mutants are now able to have a quite frequent activity, and it responds a little bit to acidosis. So similarly to SAHA, you can see that after 8 hours, treating the preparation with this drug is clearly improving breathing activity with an increase in the basal frequency and a restoration of the response to acidosis. So we have a second molecule that is doing the same, but then with this one, we went a little bit further, try to decipher the mechanisms of action. How can this molecule increase the frequency or restore the chemoception of the preparation? So the first question was, how does this molecule act on the pre-Böttinger complex? If you remember well, I told you that this network is spacing the rhythm. So we suspect if there's an increase in the frequency that it could be on that at that level. To do so, we use a different preparation, which is actually just a slice from the brainstem. This allows to have only one of those networks rhythmically active, getting rid of the influence of the others. On this preparation, we can put an electrode and record the spontaneous activity of the network. And you can see that in the wild type and the FOX2B mutant, the preparation is able to spontaneously generate rhythmic activity. Now, what is happening when we export the preparation with to 70 DMAG? And as you can see, there is indeed an increase in the frequency that we have quantified here on this graph, where you can see that after 8 hours, you have an increase, a huge increase in the frequency. But this is not phenotype dependent because the same effect is seen for the two phenotype. Anyway, we suspect and I think we can say clearly that 17-DMAG is acting on the pre-Böttinger complex and is inducing an increase in the frequency. Now, the second question was, how does this molecule can improve the acidosis response? To do this, we use a slightly different experiment. I hope this will work. Of course, it doesn't work. Okay, so this was a movie anyway. We are using calcium imaging. So calcium imaging is a way different from electrophysiology, but at the end, we get the same signal. It means that when the cell is active, a lot of calcium is entering the cell. And we have the possibility to include in the preparation a dye that will enter the cell. And each time the cell is active, that was shown in this movie, you have an increase in the fluorescence. So these traces show that the neurons are rhythmically active in this network. Okay, so by using calcium imaging, we investigated the effect of the molecule on the activity of this network. So here you have the wild type preparation. In normal pH, you can see these little peaks that are corresponding to spontaneous activity. When we change the pH, you have an increase in the frequency. This is quantified here with a nice significant effect. And as expected, in the mutant preparation, we were able to detect nothing, because I remind you that this network, the RT-NP-FRG, is almost completely gone in the mutant. Actually, it has been shown that there might be not more than 20 cells left, no, 20% cells left of this network in the mutants. So it cannot be sufficient to trigger a rhythm. Anyway, because we were able to see an improvement of the response to pH 7.2, we thought that maybe the treatment will do something. And unfortunately, whereas while the wild type behaves normally, the mutant, we couldn't detect anything. So we were a little bit disappointed, but that's how it is. So there's two possible explanations. The first one is that the residual cells of the RT-NP-FRG are not at the surface of the preparation, then we cannot detect their activity. And the other one is that there are other neuronal groups that are involved in the rescue of the response to acidosis. So to summarize everything I've shown you, what we were able to demonstrate is that on isolated brainstem preparation, both SAHA, which is an HTEC inhibitor, and 17-DMHG, which is an HSP90 inhibitor and HSP70 activator, are able, first of all, to restore a respiratory frequency that is close to normal via a direct action on the pre-Buttinger complex network. And second, those two molecules are able to improve the response to acidosis via a still unknown mechanism so far, but we are still working on it. So to make very short conclusions about the entire presentation we gave with Isabella. So our data have identified at least two molecules. So SAHA, also known as vorinostat, and more recently 17-DMHG, that is less toxic than the other ones, as promising drugs for the treatment of respiratory deficits in CCHS. Interestingly, these agents are currently already used to treat other diseases, either fully established or still in clinical trials. Of course, what is missing so far is the in vivo investigation of the effects of this molecule onto the animals. So we are working actually with the Gallegos Matros lab in Paris to try to get those data as soon as possible. But I would say that the most important message is that it seems reasonable to think that we can at least alleviate and maybe treat significantly the respiratory anomalies by the use of pharmaceutical molecules. And I will end by, as Isabella already did a bit, thank everyone who participated. The big Italian mafia from Isabella's lab, and especially Chiara Africano, who did her PhD work on this. A big thank to the Atmosphere startup created by Xenia Proton de la Chapelle with a strong involvement of Nathan in this story. Also Tiziana Bacchetti, as Isabella said, and some people from France. There's not only Italy, but also France, and especially Gabriel Pitola, who did a PhD financially supported by Atmosphere in my lab together with me, and Amelia, who is a member not only of my group, but of the group of Gallego Matros lab, and of course, all the association. And I would like also to thank you for your attention and for the invitation. Thank you.
Thank you, guys. That was very interesting. Thank you for sharing. Do we have questions? Oh, I see some hands. So I'm gonna start with Dan. You have your hand up? Go ahead.
Thank you. I'm pushing all kinds of buttons here. Great talk. I'm curious. So it sounds like the two drugs you tried are probably not causing a proliferation of FOXP2B RTN neurons. They didn't rescue the pH response, at least the 17-DMA-AG did not. I guess my question is, do you think if you administered it either earlier or longer, do you think you could actually cause a proliferation of the RTN neurons?
Well, the point is that in the literature, it's said that the neurons are not there. So I'm not sure that you can create some FOXP2B neurons using those molecules.
If you did it early enough, perhaps you could rescue their differentiation, is what I'm wondering.
Yeah, maybe, but then yeah, we would have to start very early during the embryogenesis. Maybe we didn't try it yet, but yeah, could be a good point.
Thank you.
James Oakley.
Sorry, hey there. Just a quick question. In terms of thinking about moving it on, or let's say using different compounds which might be more amenable, especially to young children, would it be a consideration to use something like how SAHA came out of something similar but less toxic, something like valproic acid or something like that, which is a milder version that has less side effects to actively test in patient, let's say?
Isabella, do you want to answer? I didn't.
I didn't hear very well.
Yeah,
Actually. Yeah, you
Yeah.
too. Yeah.
Okay.
Sorry, I'll move the laptop close to my face. I've got some background interference. Would it be possible to use a milder version, something which, again, would have similar effects, let's say to SAHA or something like that, to test earlier in, let's say, in humans or in mice, essentially, to see if it has a similar effect but with less side effects?
Okay.
Isabella, I think he's asking if there's maybe
Yeah.
drugs that have less toxicity to them.
Yes. There are many, as far as I know, there are many HSP90 inhibitors available, and also many HDAC inhibitor like SA and 17-DMAG. But there are not many those who are which are already on clinical trial or already in the market, and these two are among these. So despite the fact that they have a number of secondary effects that should be evaluated, of course, on a clinical base, I think these are two molecules that each of them in the class they belong to are among the most I would say suitable, maybe. I don't know. I'm not a pharmacologist, so I mean I'm answering without having a title to do that, but that's just a reasonable reasoning.
Isabella is a geneticist, right?
Yes.
Isabella?
I'm a geneticist.
Go ahead, Anna.
No, that's great. Thank you.
So I'm going to try to ask a question, and if this is outside of a realistic thing that you or anyone can answer, then that's fine. I know in some of the literature with non-polyalanine repeat mutations, there is like a potential understanding for the mediated decay of the protein. So I feel like what you guys are doing is kind of mimicking that in some ways, because it's degrading the aggregated proteins that come from the polyalanine repeat mutations. But my question would be that, you know, you did a lot of really good things. You showed a lot of really promising stuff, but not only does the condition impact the breathing, but it also impacts a lot of different things. And so would the degradation of the aggregated protein that you're doing, and you know, is it some in your plan, I guess, in your research plan or in someone's research plan to look at like the impact on heart rate variability or orthostatics, I guess, like various impacts on that, or hormonal deficiencies, or how that could, like with the, I guess, repair, or maybe not repair, but with the degradation of the aggregated protein, how could that impact those types of things? Or do you have that in your head?
Shall I try to answer then? Yeah. If I understood well, you're asking about non-polyalanine repeat mutations.
No,
Yeah.
I was just kind of asking more about, it seems like what you're doing is similar to what the mediated decay would be doing, which is like reducing or not having the aggregated protein. But I was just curious. I was more curious about what the impact would be on other systems than just the breathing. Like, do you have that in your plan to test that on some of the mice that you're looking at?
Some of the other phenotypes associated with CCHS beyond breathing. Yeah.
Yes.
Good question, Anna. Anna's a patient.
I don't know if I'm answering in correctly, but yes. we in vitro we concentrated on aggregates because this is a marker that we can follow easily, but so far there is no evidence that aggregates are also present in vivo. So aggregates are a powerful in vitro marker that indeed helped us to identify some molecules that afterwards resulted pretty interesting. So that's something that we believe on and we trust. But still, we don't know if aggregates are the real reason why the neurons are not there where they must be, as Muriel said, during the mouse fetus. So I don't know if I answered, because yes, I may not. I may not have gone right away to the questions.
Two more questions, I think, and we'll have to move on. But Debbie? Oh, Debbie, you're muted.
So we'll start again. Both of those presentations were amazing and inspiring. I actually have a pretty basic question. So when you're showing the increase in frequency in the isolated preps, in humans, we can hypoventilate in one of two ways. We can have a slow respiratory rate and a decent tidal volume, or it can have a diminutive tidal volume and a decent or even rapid respiratory rate. And Michael Carroll showed 11 years ago that in response to asphyxial stimuli, 14% oxygen, 7% carbon dioxide, that respiratory frequency went up, not adequately in patients with CCHS, but it went up, so there was some response. If you're totally focusing on respiratory frequency in the models, is it possible that they're still hypoventilating? They just happen to have picked up their rate.
Yes, absolutely. What we did was on a very minimal piece of tissue with isolated respiratory networks. So this is what I've talked about is the command that is sent via the phrenic nerve to the diaphragm. But then in the whole animal, many other things are, of course, contributing to the breathing activity. So that's why we definitely need to do the experiments in vivo on the animals to see whether there's still hypoventilation or whether it's a real acceleration of the rhythm. So we need in vivo experiments, that's for sure.
Thank you.
Thank you.
Linda, go ahead.
So SAHA is an HDAC inhibitor. And when I think of that, I think of transcriptional effects. And so you have somewhat of a mechanism with the decrease in HSP70. My question is, do you think there are transcriptional effects of SAHA? And if there are, how specific might that be to the activity in the Pre-Botzinger complex?
Okay, so I will answer the second part of the question.
Okay.
And I leave the first part to Isabella.
Okay. Yes, okay. So I will start. Okay. Yes, for sure. It is reasonable to imagine that the SA has an effect on expression regulation. That's definitely likely. How specific it is, we don't know, and it may have an effect on FOX2B itself, or or some target FOX2B genes, or even on chaperones, and this is more likely, of course, that
Right.
that have an effect on also the chaperones. Yes, would be interesting that to have a transcriptomic analysis. I mean, yes, we had it with SA. We did it. We did the transcriptomic analysis with 17AG. We could do the same with SA.
Yeah, good idea.
Yes. Thank you.
And now to answer the second part of the question, the action of the drug onto the pre-Botzinger complex, I don't think it's through a transcriptional effect. We actually rather suspect a general effect via microglia, which is a completely different topic. And I have no time to explain everything to you, but this is where we are going. So it's not a transcriptional effect, at least on the pre-Botzinger complex, we think.
Thank you.
There are 3 additional questions in the chat. I don't know if we'll have time to answer all of them.
I think the one is rather like, there are great questions in the chat. If you guys can maybe go and look at that, Isabella and Muriel, but there is one question about like the patients. How big is the step for human CCHS patients to take advantage of this research? And what do you think of the effect would be the effect in CCHS patients using treatments with this drug? So looking at the bigger picture, like how long would it take to maybe test this in a patient population? And no one's going to hold you to it. Because it takes a while to develop these things from this stage. And so we're probably talking multiple years off, like 10, 15, probably. I don't want to put words in anyone's mouth because these things do go slowly. And of course, the patient population would like to see this stuff go faster to have an effect. So anyone want to comment on that? That came from Patrick. It's hard. I know it's hard to predict. Researchers don't want to do this. But.
Yes. I cannot. I will start. I cannot predict, definitely. And in vivo studies is definitely needed first of all, and based on the results, then we might make a step forward, but well, you don't know until you do that, and even after that, you have to wait carefully pros and cons of treating with a molecules that has anyway some secondary effects. Yeah.
So there are more questions in the chat. Maybe look at one's from Casey and one's from Luo Shao, I think. But anyway, we do have to move on, unfortunately.
So maybe
we appreciate this. Thank you so much. But go ahead, Avatar.
Maybe Isabella and Muriel can try just to answer in the chat these 2 questions, and we will proceed with the next presentation.
Okay.
Thank you.
Okay. Thank you so much. So our next speaker is Dr. Luis Hernandez Miranda, and Dr. Hernandez Miranda is a principal investigator